Good evening to all the students of IPE by PC stream. So yes, today we are going to start on the long answer type of questions which are very important. Definitely you will get a question from these two chapters. So those two chapters which we are going to discuss today are strategies for enhancement in food production and sewage treatment from microbes in human welfare. These two are the major chapters from which definitely a long answer type of question is asked every year in the IPE paper. Okay. So this is for second year students ma. So here if you see describe tissue culture. Tissue culture technique is widely asked question many a times asked question ma. Tissue culture from strategies for enhancement in food production. What are advantages of tissue culture or conventional method of plant breeding in crop improvement programs? Okay. So advantages of tissue culture we have to mention. What are the advantages of the tissue culture in comparison with the conventional method of plant breeding? And first thing is you have to describe the tissue culture technique in detail. So here is the sequential description of the answer. I will tell you on how you have to describe the answer in a very accurate manner. Okay. So here if you see, the question has appeared in multiple years like May 2024, February, March 2024, 23, 20, 19, 18, 17 and 16, 15. So in every consecutive year you can see a question on tissue culture technique. That important it is beta. There is no point of leaving this question at all in your IP preparation. So here if you see, first you have to explain what is tissue culture in detail. Okay. What is tissue culture? Tissue culture is the technique of growing, maintaining, multiplying plant cells, tissues or organs under sterile conditions. Okay. Under sterile controlled in vitro conditions by providing the nutrient medium or in the nutrient medium. This is the accurate description of what tissue culture actually means. It is widely used in what? Micropropagation. It is widely used in micropropagation for producing genetically identical and disease free plants. Why they are mentioned as genetically identical and disease free plants? They are genetically identical to their parental plants, okay, and they are disease free as we are propagating them in the sterile conditions. The description or definition of the answer is very important in the beginning of the slide ma. Next, steps in tissue culture. First, if it is better if you first write down the steps and then explain each step in detail. What are the steps of tissue culture? First is preparation of explant, second is preparation of nutrient medium, and third will be sterilization of both nutrient medium and the explant which we are taking for propagation, and inoculation, incubation, regeneration, that is organogenesis and somatic embryogenesis. Seventh step will be acclimatization and transfer. These are the 7 steps. First you mention it in the sequence, then it will be easy for you to give it out a proper explanation for each question. So here if you see ma, steps in tissue culture technique. First, preparation of explant. Any living part of the plant, any living part it might be, it can be stem, leaf, root, meristem. When it can be selected, that selected part of any plant is called as explant. Usually we will take the living part of any plant which has the desirable characteristics. Okay. Surface sterilized with sodium hypochlorite or ethanol. You have to highlight this when you are explaining, okay, because examiner will be looking for it. How we are sterilizing using what chemical? That is sodium hypochlorite or ethanol to remove the contaminants. And then preparation of nutrient medium. How a nutrient medium is prepared? This contains essential nutrients, vitamins, growth regulators. Growth regulators includes what beta? Auxins and cytokinins. These are the growth regulators, and carbon source like sucrose. Okay. These are the essentials for preparation of nutrient medium. And Murashig and Skoog medium, MS medium we will call it as, okay, is most commonly used as nutrient medium. Okay, MS medium is most commonly used as nutrient medium. These are the first 2 steps ma. First is preparation of explant, second is preparation of nutrient medium. And third step, if you see, is sterilization. Okay, sterilization here, culture medium and glassware both are sterilized by autoclaving at 121 degrees Celsius. Okay, at how much pressure? 15 psi pressure, and in what temperature? How much time it is sterilized? 15 to 20 minutes. Accurately you have to mention at what pressure conditions we are sterilizing, at what temperature we are sterilizing, and for how many minutes we are sterilizing. Okay, culture medium and glassware both are sterilized by autoclaving at 121 degrees Celsius, 15 psi pressure for 15 to 20 minutes. Okay, this prevents microbial contamination. Okay, this will prevent microbial contamination. Next, fourth step is what? Inoculation. Sterilized explant is aseptically transferred to the culture medium. Okay, sterilized explant is transferred to what? Culture medium. Done in laminar airflow chamber. This inoculation method is done in laminar airflow chamber to maintain sterile conditions. This is to maintain what? Sterile conditions. After inoculation, what we will do, ma? Incubation. Okay, incubation wherein cultures are kept under controlled temperature, light, humidity for how many weeks? 3 to 4 weeks. Okay, for 3 to 4 weeks they are kept under controlled temperature, light, humidity. Okay, next, explant divides. Repeatedly explant will divide to form mass of undifferentiated cells called as callus. Okay, repeatedly the explant will divide and this will give mass of undifferentiated cells called as callus. Next, you see, ma, regeneration. Regeneration also known as organogenesis and somatic embryogenesis. Here organogenesis, you see here callus differentiates into shoots or roots under auxin cytokinin balance. Okay, organogenesis, here callus. What is callus? Undifferentiated mass of the cells are called as callus. Callus will start differentiating in this step, which is organogenesis or somatic embryogenesis. What happens, beta? Callus will start differentiating to organs based on auxin cytokinin concentration. If more amount of auxin is provided to the callus, is provided, okay, root systems will be developed, roots will be developed first. If more amount of cytokinin is produced, they will be differentiated to stem first as such. Okay, so organogenesis differentiates into shoots and roots under auxin cytokinin balance based on that. Next, somatic embryogenesis, if you see, callus produces embryo-like structures. These embryo-like structures are called as embryoids. Okay, callus will produce what? Embryo-like structures called as embryoids that develop into a whole plant. Clear, right? So here organogenesis where callus will be differentiated into organs. This requires auxin cytokinin balance, and later in somatic embryogenesis callus will produce what? Embryo-like structures called as embryoids. So this is how. First, what, ma? We will select the explant. Second, preparation of nutrient medium. Third is sterilization. Fourth will be inoculation. Fifth is incubation. Sixth, regeneration, which can be organogenesis or somatic embryogenesis. And last step is acclimatization and transfer. The plantlets which are produced after organogenesis, small plantlets which are produced, these plantlets will be transferred to the greenhouses. The plantlets are transferred from the culture tubes to greenhouses and finally to the field. Okay, they are transferred from culture tubes. They are transferred from culture tubes to greenhouses. Okay, and finally into fields. Advantages of tissue culture over conventional breeding. Okay, advantages of tissue culture over conventional breeding. What are the advantages of tissue culture? What is conventional breeding? Traditional breeding. What are the advantages of tissue culture over traditional breeding? Here you see, first is mass propagation. You can take a small explant and you can propagate hundreds of plantlets from a small explant. So, the first benefit, that is what? First benefit is mass propagation. First benefit is mass propagation. Large number of plants can be produced within a short time and small space. This is micropropagation. Second is genetic uniformity, okay, where plants produced are genetically identical to the parent plants. Okay, they are genetically identical to the parent plants. These are called as clones, somaclonal plants. Third, disease-free plants. Okay, disease-free plants, where meristem culture ensures elimination of virus. Okay, disease-free, this happens because of what? Sterilization. Sterilization, wherein meristem culture will ensure elimination of viruses, pathogens, producing what? Healthy plants later. Next, conservation of germplasm. Okay, conservation of germplasm. This helps to preserve rare, endangered and economically important plant species. Okay, this helps to preserve rare, endangered and economically important plant species. Clear, right, ma? Next, faster crop improvement. Useful in propagating plants which are difficult to breed by conventional methods like apple, banana, bamboo and orchids. This is fast crop improvement. Okay. Next, if you see year-round production. Okay, what is year-round production here? Independent of seasonal constraints enables rapid supply of planting materials. Okay, independent of seasonal constraints, this enables rapid supply of planting materials. Okay, this is year-round production rather than producing plants in different seasons. We can produce, with the help of micropropagation, we can produce plants throughout the year. Next, artificial seeds. Somatic embryos can be encapsulated to form synthetic seeds. These synthetic seeds can be stored easily. Okay, they can be stored easily and they can be transported easily. These are the major benefits of what? Tissue culture. Okay, so to remember the tissue culture in detail, here is a flow chart which will give you a detailed analysis on this. It is up to your comfort, ma. First you can provide the flow chart and then you start on explaining each step of the tissue culture. If not, first you explain, then you give the flow chart. It is completely your comfort. But then in the long answer question of tissue culture, you have to mention the flow chart, you have to mention the sequential description of each step, and lastly, how tissue culture is more beneficial than conventional breeding method. Okay, these three things you have to mention in a sequential manner. Here if you see the flow chart showing plant tissue culture technique. First is what, ma? Preparation of medium and getting the explant. Okay, after which sterilization is done. This is the third step. And then inoculation followed by incubation. Incubation is followed by callus formation. Callus formation, then based on auxin cytokinin balance, this gives what? Organogenesis and somatic embryogenesis, and later they will be transferred to the greenhouse gases or greenhouse glass. Okay, so here if you see the sequential steps, I will write it here. Okay, it will be easy for you. So what are the steps, ma? First is selection of the explant. Second will be preparation of medium. Third is inoculation. Fourth is incubation. Fifth, callus formation. Sixth is organogenesis. And seventh will be transfer the plantlets to greenhouse glass. Fine, right? These are the sequential steps of what? Tissue culture technique. Okay. Next question, which is very much important after tissue culture technique, is You are botanist working in the area of plant breeding. Describe various steps that you will undertake to release a new variety. Okay, what are the steps that you are undertaking to release the new variety? These questions have asked in March 2018, 2017 and 2016. Here if you see, answer: plant breeding and development of new plant varieties. Okay, what are the varieties of plant breeding? Okay, what are the new varieties that are produced from plant breeding, and what are the steps of plant breeding? Here you see, first define what is plant breeding. Plant breeding is the application of genetic principles to produce desired crop varieties. Okay, to produce desired crop varieties with improved yield, quality, resistance to diseases and tolerance to stressful conditions. To release a new variety, a series of systematic steps must be followed. So, first describe the plant breeding in detail in 1 or 2 sentences and then start on giving out the steps of plant breeding. So, here if you see, steps in breeding a new genetic crop variety. So, here if you see, first collection of variability or germplasm connection. Okay, connection of variability or germplasm connection. Here genetic variability is foundation of any breeding program where wild relatives, land races and existing cultivars are collected and preserved. Okay, whatever you are describing, you can describe in the proper terminology in your own pattern, but then give it out in a point wise manner. Paragraphs are not encouraged at all. Okay, don't give out your answer, long answer type of question, don't present it in the paragraph wise pattern. Always present the answer in the point wise pattern only. This will attract the examiner to give you more marks. Okay, so this pool of genetic material, that is germplasm, this will provide raw material for breeding. For example, here if you see, disease resistance genes from wild wheat varieties. This is one example for collection of variability, that is germplasm collection. Okay. Second is what? Evaluation and selection of parents. Germplasm is screened to identify plants with desirable traits such as high yield, disease resistance and nutritional value. Okay. The selected plants are then multiplied and they are used as what? Parents in hybridization. They are used as parents in hybridization. Pure lines, okay, pure lines which are homozygous are often developed to maintain what? Uniformity of the pattern. Okay. Third, after evaluation and selection, what is the third step that we take, ma? Third step is cross hybridization among selected parents. Okay. Cross hybridization among selected parents. Here desired traits from 2 different parents are combined through cross pollination. For example, if you see, a disease resistant plant is crossed with a high protein quality plant. Okay. Disease resistant plant is crossed with high protein variety plant. Male parent pollen, let us consider male parent pollen, which male parent is disease resistant on the stigma of the selected female flower. Female parent is high quality, is having high quality of protein. Let us consider this way. Though thousands of crosses are done, only a few hybrids exhibit a desired combination of traits. Though thousands of crosses are done, only few hybrids will provide the desired traits of combination of traits. Next, fourth is selection and testing of superior recombinants. Okay. Hybrid progeny are evaluated for desirable characters such as what? Yield, resistance and quality. Superior plants are selected and they are subjected to repeated self-pollination to obtain uniform stable lines. Fine. Scientific evaluation will ensure only best recombinant plants which are advanced. Next, last step is of course what? Testing, release and commercialization, where we will test whether both the desirable characters are present in the offspring or not, and then we will release them and then we will start cultivating the plantlets which have both the desirable characters. So, here if you see, Newly selected lines are grown in experimental fields under ideal conditions. They are tested across different locations and seasons. Okay, they are tested across different locations and seasons for at least 3 years. For at least 3 years they are tested. Performance is compared with existing cultivars with reference to check varieties. If superior, the variety is officially released and made available to farmers for large scale cultivation. Okay, these are the sequential steps that you have to write it, ma. First, after you do one thing, first after writing down the definition of plant breeding, keep the heading of steps in breeding a new genetic crop variety. Here itself you give out the headings that collection of variability or germplasm connection, second evaluation and selection of parents, third cross hybridization among selected parents, fourth selection and testing of superior recombinants, fifth testing, release and commercialization. These 5 steps you mention first, after which each step you start briefly elaborating each step. Okay. Next, this flow chart. What are the main objectives of plant breeding? Here we go. This is a creative way of presentation of the objectives of plant breeding. You can also give it out in this way. Here we go, objectives of plant breeding. First, higher yield, second, resistance to diseases and pests, third, change in maturity period, fourth will be elimination to toxic substances, synchronous maturity, photoinsensitivity, resistance to abiotic stress and improved quality. These are the points to be highlighted under objectives of plant breeding. Clear, right? Yes, ma? Yeah, next. Microbes in human welfare. So, those 2 questions, one is about tissue culture, second is plant breeding. Most important questions from strategies of food production and enhancement. Now, the only question that we have to focus from microbes in human welfare is sewage treatment. Highly important beta. Sewage treatment, where we have to focus on the primary sewage treatment, secondary sewage treatment, in that also aerobic, anaerobic modes of sewage treatment. This sewage treatment has appeared in the years like March 23, May 2022, August 2022. These are the 3 consecutive years wherein this question has appeared. So, here if you see the answer, ma, microbes in sewage treatment. Sewage is a waste water from households and industries containing what? Organic matter, pathogens and other pollutants. Before releasing into the rivers, streams, sewage must be treated in a sewage treatment plant, that is STPs. Okay? Sewage treatment plants, this sewage has to be treated. This treatment mainly uses heterotrophic microbes present in the sewage. Okay? They will use heterotrophic microbes from the sewage which are present in the sewage. How it is being done? First you give out a flow chart beta. First you draw out the map on how you have to conduct this question, then it will be easy for you to explain this question. First, the wastewater influent or sewage materials are first passed on through the primary treatment. Primary treatment includes sequential filtration. This includes sequential filtration and sedimentation. Sequential filtration and sedimentation. Then primary clarifier, then it will be passed on to aeration tank. After then secondary clarifier, which is anaerobic sludge digester, and then effluent is passed down to the water bodies. This is the sequential flow that you have to remember. and then you start on explaining each thing, primary treatment, secondary treatment in that aerobic and anaerobic. Here we go, sewage treatment is carried out in two main stages. First, primary treatment or physical treatment, which involves mechanical removal and small particles, mechanical removal of large and small particles. For example, here you see the steps which include sequential filtration. This removes what? Floating debris. And sediments, what type of sediments are removed, like grit, soil, pebbles that will be sedimented at the bottom after sedimentation. So, sequential filtration will remove floating debris, sedimentation will clear off grit, soil and pebbles that are present in the bottom, where in the sedimentation tank they will be settled at the bottom. Now, the primary effluent, here in this sedimentation tank, the dirt which is settled at the bottom is addressed as primary sludge, and the water particles which are above this sludge, these are called as what? Effluent, primary effluent. This is primary sludge and this is primary effluent. The settled solids form primary sludge, while the liquid above the primary effluent. The effluent is then passed for secondary treatment. Okay, secondary treatment or biological treatment here. Okay, secondary treatment or biological treatment. Here you have to pass on the primary effluent first to the aerobic sludge digesters, second to the anaerobic sludge digesters. Aerobic sludge digesters, if you see, aerobic treatment, where primary effluent is introduced into aeration tank and then continuous agitation or pumping will be done. This will encourage growth of what? Aerobic microbes. Microbes will grow into flocs. Flocs will have what? Bacterial masses and fungal filaments. Okay, bacterial flocs will be having bacterial masses and fungal filaments. Fine, right? And here you see these flocs will degrade organic matter and they will reduce biological oxygen demand. Reduction of BOD is a strong indication of lowering organic pollution and making the water safer. Now, here from this the effluent is passed on to next what? Anaerobic sludge digester. After BOD reduction, okay, reduction in BOD, and here you can also include the point that increase in dissolved oxygen, increase in dissolved oxygen. Fine, right? A part of this sludge is recycled back again to the aeration tank. This serves as what? Inoculum. Okay, this serves as inoculum. The remaining sludge is sent to the anaerobic sludge digesters, where anaerobic bacteria and fungi will decompose the organic matter. This produces biogas. Biogas will have what here? Carbon dioxide, methane and H2S. This biogas is inflammable. Okay, this biogas is inflammable and this is used as what? Energy source. Next, finally, treated effluent is released into the rivers and streams. Significance of microbes in sewage treatment. What is the significance of microbes in sewage treatment? This is the last way we are explaining what? Sewage treatment. Reduce organic pollution. Sewage treatment will reduce the organic pollution. Microbes lower BOD and purify water. Microbes will lower biological oxygen demand and this will purify water. Biogas production, here if you see, anaerobic microbes will generate useful biogas as byproduct. This is safe water disposal. And the treated effluent is less polluting when it is discharged. Eco-friendly method. Here we are not using any process which harms the environment. So, this is an eco-friendly method. This utilizes what? Natural microbial activity, natural microbes, heterotrophic microbes which are present in the sewage, okay, wastewater. Okay, this is how you have to present. Sewage treatment, first you draw the flow chart and then explain about primary treatment and then about aerobic sludge digestion, after that anaerobic treatment and what you are producing like biogas at the end. Okay. After that, significance of the sewage treatment. Okay. After which you have to highlight what? Significance of sewage treatment. With this, in today's class we have dealt with 3 long answer type of questions which are very important, most expected questions this time. First is what? Tissue culture. Second is plant breeding, and third is what? Sewage treatment. These are the 3 questions that are done for today. In our next class, we will be starting with long answer type of questions from biotechnology. Okay. So, that is all for today's session, students. Thank you.